recorded. All right, from the top. Here we go. Brumfeld is here. Champion is here. Gorby is not here, but Gross is now. All right, yeah, Gross is here. Gunther's here. Gunther is here. Both Hills are here. Jackson, not here. Keys is here. Perez is here. Sigmund is here. Slaughter is not here. Smith is here. Stover is here. Tompkins is not here. The Nest is here. Watson is here. Young is still here. Lieback still here. Lawrence still not here. Wilson still not here. And that's it. Did I miss anybody? All right. I am reporting this because the people who are not here, still hoping they watch this, they won't get the full 20 points. Excuse me. They won't get the full 20 points that you guys are getting for being here on time. If you're watching the video, it's the same as the usual things. You're going to send me the attendance words, with screenshot every time I say an attendance word. And in your case, you're going to get 10 points for today's lab instead of 20. However, there is an online version of this lab. If you do this online version, this goes for everybody. If you do the online version of this lab before we do the actual version next week, you can get 20 points of extra credit that'll be applied to your lab. For those of you who are not here, 10 of those points can be added to this, so you can get your full 20 points for this week's lab. But again, you're going to have to jump through some extra hoops to get those points. So any questions about that? All right, for the rest of you, unlike when it's a normal lecture, there's really no need to send me the attendance words because there's no extra credit to give you. The most points I can give you per week is 20 points, right? So nothing I can do. So no need to do it. Anyway, let's get started. I'm trying to make this quick, but it's, I just want to make sure you truly understand it. And also, again, obviously, since I'm recording this, you can always go back and watch it. So 3 weeks from now, when you're writing your lab report, like, man, what did he say about that? Well, you'll be able to watch the video and remind yourself. But of course, I'm also here to help you. Anyway, so yeah, like I've already mentioned, today's lab grade is based on attendance. If you're here, you get the full 20 minutes as long as you stay the whole time. I'm gonna get rid of that one. Half credit's not true. But I don't know. I'll deal with that later. Next week is going to be like normal, right? You have 5 points for the pre-lab. This is due this Friday. You're going to have 15 points for the lab worksheet that's going to be due the following Friday or past due the following Friday. So that part's normal. The only new part is you also have a 20-point blackboard that's going to be due. So in a sense, this is the most important lab as far as grades are concerned. It's the most important lab because it's worth double the points. Almost triple when you think about it, because today you get 20 points for just being here, then you get 20 points for the actual lab, and then 20 points for the lab report. So as far as grades are concerned, this is a very important lab. So, all right, I was just mentioning, if you come in late, you only get half points, but there's a video you can watch. I'm recording this, so if you watch the video, you can get extra points, and there's an online version of this lab. You can do the online version and make up extra points. Email me if you have questions. I'll explain it to you later, but I want to get this ball rolling. So here's the questions we're going to ask and answer in next week's lab. The first one we're going to ask is, does the rate of photosynthesis increase as light intensity increases? So in other words, as we add more light to photosynthetic organisms, will they photosynthesize more? Right? That's the first one. The next one is, do photosynthetic organisms respire when light is absent? So that next question, don't let the photosynthetic part throw you off. We are talking about photosynthetic organisms, but this next question is all about respiration. Like, will these things do respiration in the dark? That's what that question is. The next one is, does an increase in temperature result in an increase of photosynthesis, right? So as we warm these things up, will they do more photosynthesis? Will they do less photosynthesis, or will there be no effect? And then kind of the opposite of that is this next one. Does an increase in temperature result in an increase in respiration? So again, in that case, we're trying to see how changing the temperature changes the rate of respiration. And then finally, the last one we're going to look at is, which colors of light result in an increase in the rate of photosynthesis, right? So we're going to have all these photosynthetic organisms under all these different colors of light, that we're going to see if that affects how quickly or how much they respire or do photosynthesis. So how can we answer these questions? That's where we're coming to next. How can we answer them? Obviously, we're going to, well, actually, let's see if anybody gets this. Man, that's too vague. Let's say this. What we need to do is measure the rate of photosynthesis and respiration, right? That's the data that we need. This is what we're seeing, right? We're trying to do all these different things to these photosynthetic organisms to see if it changes how fast and how much they photosynthesize, how fast they do respiration. That's the first word for attendance. For those of you watching the video, measure is the first word for attendance. So the question is then, how? Right? So if this wasn't a lab that we're doing in an hour and 50 minutes, if we had all semester to do photosynthesis, for example, how could we measure photosynthesis? If we had all semester and we could put some plants under some different colors of lights for all 16 weeks, what measurements could we take to see how much they did how much they photosynthesized? This isn't a trick question, so don't get too, don't think too hard into it. It should be a pretty easy answer. There's a few different answers.
How big they get.
Yeah, how big they get. I like that you said that because that could be, we can take that a few ways. How tall do they get? How wide do they get? How many leaves do they produce? Their circumference, how does that change? How does their weight change? Anyway, so yeah, that's one way we can do it, right? But obviously we don't have all semester. So we had to come up with a different way of measuring photosynthesis and respiration. The question is how, and that's what we're going to talk about really quickly. So first of all, let's look at the equation that you need to know for the exam, and obviously you're going to need to know for the lab too. The basic equation for photosynthesis, minus all the numbers, if you add water and carbon dioxide, you get glucose and oxygen. So that being said, we wanted to measure photosynthesis within 2 hours, about 2 hours. We can measure any of these things, right? To determine how much it's photosynthesizing. For example, if it's using water, does that mean it's photosynthesizing or respiring? Right? If it's using water, does that mean it's doing photosynthesis or respiration? It's right there in front of you. Yeah. That means it's doing photosynthesis, right? Photosynthesis, you can see in this equation, uses water. So if we can somehow measure the water levels, if the water levels go down, we would know it's doing photosynthesis. Likewise, if the carbon dioxide level was going down, we know it's doing photosynthesis because photosynthesis uses carbon dioxide. If we could somehow measure the glucose levels, if they were increasing, we would know it's doing photosynthesis because photosynthesis produces glucose. If we could somehow measure the oxygen content, if it was going up, we would know it's doing photosynthesis, right? Because photosynthesis produces oxygen. Of course, you have the opposite. The opposite is true of respiration, right? Respiration uses glucose and oxygen to produce carbon dioxide and water. So obviously, the opposite will be true here. If we can measure the levels of glucose, if they're going down, that means respiration is happening. If we can measure the levels of oxygen, if it's going down, that means respiration is happening. Carbon dioxide levels are going up, respiration is happening. Water levels are going up, respiration is happening. Does that make sense to everybody? It's some basic stuff here, right? We're using things, we're producing them. So if we can measure any of these things, we can tell whether they're increasing or decreasing, which of these two processes is happening. So here's what I haven't taught you though. When you mix carbon dioxide with water, you get something called carbonic acid. Acid, it's in the name, right? It's acidic. Therefore, where are we at? Photosynthesis, as you already know, reduces the level of carbon dioxide because it's using carbon dioxide, right? And if it's taking photosynthesis, taking carbon dioxide out of the equation, that means there's going to be less carbonic acid. Hopefully that makes sense, right? So if you mix carbon dioxide and water, you get an acid. Who cares the name of it? Yeah, it's carbonic acid, but who cares? You mix carbon dioxide and water, you get an acid. If photosynthesis is happening, you're reducing the levels of carbon dioxide. Therefore, you're also reducing the levels of acid. So what should the pH do? If you're making it less acidic, which way is the pH going to go? Up or down? Making it less acidic. Man, I hope you guys are just shy. Yes, thank you. Up, right? Because basic is up, acidic is down. Therefore, when we do these experiments, I'm going to get more details here in a second. we're going to test, we're going to be able to monitor the pH. If the pH is going up, that means photosynthesis is happening. I want to pause and make sure you understand that most very basic concept, because you have to write about it in the lab report, which again we'll talk about later. If the photosynthesis is happening, pH has to go up, right? Because you're removing this carbon dioxide, which essentially means you're removing acid. Obviously, the opposite is true for respiration. As we're measuring this pH level, if it's increasing, that means we're producing more acid. And we're producing more acid because we produced carbon dioxide, which is mixing with water. So pH is going down, that means respiration is happening. And again, I'll pause. This little part right here is so important. You need to know that to understand how these experiments are going to work. Photosynthesis is happening, pH goes up. Respiration is happening, pH goes down. Keep that in the back of your mind for this entire lecture and for the lab. Now, that being said, let me get a little technical. Not that you need to know this for the exam or for the lab, but technically, when I say, for example, pH goes up, that means photosynthesis is happening. A more accurate way to say it, you don't need to worry about this, but just so you know, technically that just means photosynthesis is happening more than respiration. It's technically, probably both of these things are happening at the same time. So if you're giving it a lot of light, it's probably going to do photosynthesis, even though it's still doing respiration in the background, and that's why there'll be an increase in photosynthesis. So yes, for the lab, you need to associate an increase of pH with photosynthesis and a decrease with respiration, just so you know to keep in the back of your mind. What's really happening is that it's an increase. That means we're just happen to be doing more photosynthesis than respiration. If there's a decrease, that just means we're doing more respiration than photosynthesis. So any questions about this slide? All right, trying to get through this quickly. So how are we going to measure the pH? Let's talk about that. First of all, you have these things called cuvettes. They're not going to look like this. They're going to look a little bit different, but it's the same concept. It's just a little bit of little thing. It's a little thing in there. And that thing is going to have some liquid in it, right? Because there's going to be some algae in there. Algae need liquid, right? Then in addition to just the water that it needs to survive, it's also going to have a pH indicator. And you're already familiar with this because we used it in lab 2, right? We used the pH indicator to see how the pH changed. We added base and added acid to it. Same thing here. So you're already familiar with this process. You're going to have these things. You're going to have this pH color guide. And every 5 minutes, you can see here, this is basically what it's going to look like. Every 5 minutes, you're going to measure the pH, write down what the pH is. If the pH gets larger, that means photosynthesis is happening, right? The pH is going up, photosynthesis is happening. The pH goes down over time, we know respiration is happening. So that's how that's going to work. So let me ask you this, though. Why do it every 5 minutes? Why not just say, all right, at the beginning, it was this pH, and 45 minutes later, it was this pH? Why do we need to take these measurements every 5 minutes? Does anybody guess? No guesses? All right, let's make it very basic. Let's assume we were just doing the one that's about light intensity. Let's say we had one of these things filled with algae, and it was really close to a light. We had one that was really far away from a light, and we only took the measurements at the beginning and the end. And let's say the one, they both started at 7, right? Let me put a line here. Let's pretend like that's a column or 2 columns. Let's say they both started at 7 and both ended at 10. Which one photosynthesized faster or more? The one on the left or the one on the right? You don't know, right? That's why we take measurements every 5 minutes, because this might happen. In the experiments you're doing, you might start at the same pH and end at the same pH, which tells you nothing, which is why you need to look at the data every 5 minutes. Because it may go like this. The one on the left may have gone 7, 8, 9, and then hit 10 and stay there the whole time, right? This one on the right may have been 7, and finally it hits 8, finally it hits 9, and then finally it hit 10 at the very end. In this case, and I know that's really sloppy, but in this case, we can look at this data and, all right, even though they both started at the same and ended at the same, the one on the left, that one photosynthesized much quicker, right? Because it got to that pH of 10 and stayed there much quicker. Does that make sense to everybody before I move forward? All right? So, again, these cuvettes are filled with a pH indicator and algae beads. Again, these are the questions that we're going to be asking. I've already mentioned it once, so I'm going to go through this very quickly. Let's look at the first question. Do photosynthetic organisms photosynthesize more in intense light? So we're going to have a bunch of different cuvettes filled with algae beads. One of them is going to be in the dark, right? So it's not going to be exposed to any light. Therefore, we should expect it to decrease, right? Because it shouldn't be doing any photosynthesis. One of them is going to be very close to the light. Therefore, we should expect the pH to go up. One of them is going to be a little bit farther from the light, and one of them is going to be even farther from the light, right? So we're going to have three of them at varying distances. And when we get there, you'll see when the lab starts, we might call some audibles. We might change things. The lab might say, make sure that this far, this far, and this far. But these are your experiments. So whoever's doing this experiment, you can change it up if you want. Like instead of keeping them this far, this far, and this far, let's really space them out and do them this far, this far, and this far. And you'll see what I mean when we get to the lab next week. But keep in mind, all these are basic guidelines. We can make some changes if we need to. As a matter of fact, one of the things I'm going to tell the guy who gets our stuff together, I'm going to ask him if he can give me more than 4 cuvettes for this experiment. And if he can, then we're going to have an extra cuvette, like one close to the light, or I said maybe one far from the light, close to the light, close to the light, closest, right? So obviously the more data you have, the better. But anyway, that's how that's going to work. Any questions about that one? All right, the next one is very simple. Do photosynthetic organisms respire when light is absent? For that one, we're going to have one in the dark and one in the light. Technically, we could just use the data from the first experiment to see this, but we need to break it down into 5 groups so students can do their own experiments, and that's why we have this one. Yes, one's going to be in the light, it's exposed to light, therefore it should be photosynthesizing, therefore the pH should go up. And one's going to be in the dark, not exposed to light, therefore not photosynthesizing, therefore the respiration, the pH should go down. Any questions about that one? All right, the next experiment will be, does it increase in temperature? Right? When we change the temperature, does that affect the rate of photosynthesis? This one's going to be very simple too. Have one in the light at room temperature, and have one in the light on ice. Nice and simple, right? One's going to be warm, one's going to be cold. Either way, they should both be going up because they're going to be exposed to light. They should be photosynthesizing. So we're just going to see if it makes a difference. If we can, again, if I get a third cuvette, maybe we'll put one on a hot plate. So we'll have one that's warm, one that's room temperature, one that's cold, if possible. Again, we can call audibles. These are your experiments. We can change them as we want to. This is just a basic guideline. Any questions about this one? All right, the next one's also very simple. Instead of looking at photosynthesis, we're going to be looking at respiration. Other than that, it's the same. You're going to have one in the room temperature, one in the ice. It's going to be covered up, not exposed to light. Therefore, it should be respiring. Therefore, the pH should be decreasing. Any questions about that one? All right, the last one. I don't know why, but for me, I enjoy this one the most. This one, again, we're looking at respiration, the rate of respiration, but we're going to see how they do under different colors of light. At the very least, we're going to have one under a white light, one under a green light, one under a red light, and one under a blue light. And for all of them, in theory, the pH should be increasing, right? Because in theory, they should all be photosynthesizing. And again, if I can get some extra lamps, and I can get some extra cuvettes, maybe we'll throw some other stuff. Maybe there'll be an orange light or a yellow light if I can pull it off. And again, just to remind you, again, these are your experiments, our experiments. We can change them if we want. This is just a guideline. I mean, for that matter, something could go wrong here. Maybe the green light broke and we can only use white, red, and blue, right? So point is, we can change them as we want. So this next slide is kind of in reference to the lab report, even though I haven't really gotten into the lab report. I just want to make sure this is very clear. These experiments are not about pH, right? pH is simply what we're using to measure the rate of photosynthesis and respiration. It goes back to what I was talking about earlier, like if you had a whole semester to do this, and I asked what data could we take? And I think Gross said, see how big they get. And that's true, right? We could measure their height, for example. And then if we did that experiment, the experiment isn't about measuring a plant. The experiment is about how much photosynthesis is happening, and that just happens to be how we're measuring it. So keep this in mind when you're writing your lab report. pH changing is nothing but a tool. Right? We're just using it as a way of measuring photosynthesis and respiration. This is not a pH experiment. This is a photosynthesis and respiration experiment. I'm not even talking about this bullet point because we've already talked about it, right? How else could we measure it? Again, if we can somehow measure the levels of oxygen, we know if oxygen is going up, photosynthesis is happening. because it produces oxygen. We know if glucose is going up, photosynthesis is happening because it produces glucose, so on and so forth. There's all kinds of ways. Also, that will be something you need to write about on the lab report. I know I haven't officially started talking about the lab report yet, but that will be a question on the lab and a paragraph on the lab report. And I've just given you a bunch of ideas, like how else could we measure the rate of photosynthesis and respiration? All right. Just to be clear here, no cuvettes, these are the things without you, right? None of them in the dark should have an increase in pH, right? Because they're in the dark. That means they shouldn't be photosynthesizing. Likewise, I need to fix that typo. None in the light should see a decrease in pH, in theory, right? Because they should be in light. They're in light. That means they should be photosynthesizing, so the pH should be going up. So if this happens to you, know that something went wrong. There's nothing wrong with that. You're not going to lose any points. Things go wrong in experiments, but be aware of it because you're going to have to write about it in your lab report. So if something went wrong, you do need to at least guess what happened. And again, as long as you have a guess and it makes sense, then you're okay. But no one's going to lose points if it doesn't turn out the way it's supposed to. But again, you need to at least notice that it didn't turn out the way it's supposed to. And you can even write about that. And we'll get to that again. Once I officially tell you how to write a lab report, you can say at the end of it, when you're discussing your results, you might have, here's the results, but I don't know if my hypothesis is correct or not because the results are invalid. The one in the dark is photosynthesizing, so something must have gone wrong. All right, any questions about this slide? All right, this should have been a common theme throughout the semester. I've been trying to push it on you guys throughout the semester in preparation for this lab. So you should always be thinking, how can you do this in a better manner? And I don't mean the lab worksheet or how I'm teaching the class or the materials you get. I'm saying literally put yourself in the shoes of a scientist trying to publish your study, right? How can your experiment design be improved? And I've already given you some examples previously. Like when we talked about light intensity, I said if I can get more cuvettes, then we'll have more data, right? We'll have them farther away from the light. Or the one with the different colored lights, right? If I can get maybe an orange lamp. So there's some ideas, right? So you should always be thinking about this the whole semester, especially for this lab because you're gonna have to write about it in the lab report. And how can the lab experiment be improved? Not the worksheet, not the materials, but how could your experiment be improved? And also, again, like I mentioned earlier, you should be thinking about different experiments because in real science, once you test your hypothesis is supported, you don't just say you're done, you come up with a new way to test your hypothesis. So, any questions about this slide? Make a great time. The next word for attendance is forest. There's a forest. It's technically just the woods right next to the riding out with the lake. Anyway, forest is the next word for attendance. Now we're going to briefly talk about how to write a lab report. So first, you might be wondering why you have to write a lab report, right? Because none of you, as far as I know, are biologists or planning to become biologists. So, why do you need to write a lab report? There's a few reasons. First of all, it's just part of the course, right? I don't make the rules. When you have a science class, that's what they want you to do, or a biology class. The biology department dictates you need to have at least one lab report. Also, in a perfect world, I would have had you write a lab report on all the labs. Instead of doing a lab worksheet, I would have you write a lab report. Because to truly understand the experiment, to write a lab report, you have to truly understand it. Had you guys had to write a lab report on last week's lab with the whole spreading of the dye, you probably would have noticed that something went wrong there. Because you're thinking about it, you're having to write about it, so you'd be like, well, that's not right. How did the dye shrink, right? So that's another reason. And another one, and this one's really important, especially right now, is hopefully one day, as critical thinkers, you're not just going to believe whatever people tell you. So if someone comes out and tells you that Tylenol causes autism, or somebody else comes out and tells you, no, that's BS, Tylenol doesn't cause autism, either one, hopefully you do your own research. By research, I mean looking up articles. And you're reading articles. And I'm not saying one way or the other, right? Because I don't want to start any kind of political opinion. Either way, you should look at both of those sides and do some research. You should look up some articles. And to truly understand research articles, this will help you. Having written your own research article is basically what the lab report is. Hopefully that'll help you understand these research articles. So any questions about this slide? All right, here's a big one. Let's talk about some basic rules you need to follow. Do not use the internet. What I mean by that is, don't use AI to write your papers. It's getting worse and worse. When I say worse, I mean more and more people are doing it. And I teach online classes, so it's getting really bad in there. But people are getting sloppier and sloppier, and I have ways of knowing if you use AI. Don't use AI. It's a great tool, but not for this, right? Use your own brain to write this. But I did put a little star next to that because sometimes if you want to, there's some ways you can use the internet. And I'll tell you how when we get there. For now, it's just the basic rule is don't use the internet. You'll see why here in a second. Here's the good news. You're only going to write about the experiment that you did, right? There's 5 different experiments. You're going to be broken down into 5 different groups. And whichever group you're in, that's the one you're going to write about. But the lab itself is like it usually is. You're going to answer all the questions for the lab. So while you're doing it, I recommend going from group to group and talking to those people and seeing what they're doing. But for the lab report, you only have to do the one experiment that you did. Now, granted, you can do all 4 or all 5 if you want for extra credit, but those would have to be separate lab reports. We'll cross average when we get there. Just let me know if you have any interest in doing it. They all need to be past tense and in first person. So you'll say, instead of saying, in this experiment, I want to see if light intensity affects respiration. That's wrong. You want to say, in this experiment, I wanted to see if light intensity affected respiration. Of course, that's a small mistake. So, if you don't do that, you'll maybe lose like 1 point. But anyway, yeah, it's supposed to be past tense, first person. You're going to want to double space it. And this is the big question I always get. Is there a minimum? Like, how long does it have to be? There is no minimum. I'm going to show you here in a second. There's a rubric, basically. It's like a checklist. You need to have this, you need to have this, you need to have this, you need to follow that checklist. That's all you need to do. This is science. The shorter, the better. Short and sweet's the way to go. You don't want to put a bunch of adjectives in there like, first, I gingerly took the cuvette and carefully placed it under the bright light, slowly moving my hand away as I wondered if it's, you don't need to do all that. Just short, sweet, to the point. Any questions about that? Okay, independent work's no longer a thing. So you're going to turn this in the same way you'll turn it in the lab, which is, again, I'm going to give you a worksheet. You're going to write your lab report on the worksheet. You're going to save it as a PDF. You're going to upload it. It's what you're already used to. Any questions about that? All right. When does it due? That's the next big question, right? You'll have 2 weeks from the day that you get your graded lab back. So, in theory, that means next Friday. That's the past due date, right? In theory, hopefully you're all done by then, and I'm all graded by then, hopefully. So that would mean you'll have 2 weeks as of next Friday to complete it. But as you're about to see, when I teach you how to write a lab report, you do not have to wait until you've done the experiment to write your lab report. Because a lot of this is going to be a little bit of introduction stuff. Now, you obviously need your data to write your entire lab report, but some of this you're not going to need to wait. So any questions about this slide? Okay, how to write a lab report. Oh, some more stuff. So this is a continuation of the last slide. It should be free of grammatical spelling and typing errors. I'm not that nitpicky, but as I'm quickly going through it, because I've got a lot to grade, as I'm quickly going through it, if something jumps out at me, then you're going to lose a point. Right? I'm not that picky, but if you start a sentence with a lowercase letter, that's going to just jump out at me. Or if you said, I seen the PH do this instead of I saw the PH do that. Things like that jump out at me. But again, for the most part, I'm not nitpicking. This isn't an English class, so I'm not looking for these errors. But if they jump out at me, you're going to lose 1 point per error. Any questions about that bullet point? All right, here's a good guideline. It should make sense. So if you're wondering how it looks, first of all, you should come to me. If you wanted me to proofread it before you turn it in, I would love to do that. I'd be happy to do it. It's going to save you some time. It's going to save me some time. It'll probably help you grade. But it should be able to make enough sense. You should be able to give that to anybody who's not had this class, and they should understand everything that you've written in the lab report, because you should have enough background information to where it makes sense. We'll come back to that in a second. But for now, are there any questions about that bullet point? Obviously, it should be accurate and correct. There's so many ways it can be incorrect. I'm not even going to talk about that. You'll see when you turn into that report. If you write something that's right. So, for example, if you write like trying to describe what you're doing, you said, all right, so we're doing this experiment where we're putting these things under light, and we expect the pH to go down because it's photosynthesizing. That you will lose a point for that, right? Because it's not supposed to go down; it's supposed to go up. So things just need to be accurate. They need to be correct. You lose a point for every time you write something that's incorrect. It should be precise. Cross that bridge when we get there too. You probably don't need to worry too much about that. It should be succinct, like I mentioned earlier, short and sweet. Right? You're not trying to be writing this entertaining thing. This isn't a persuasive paper. You just write what you did, and that's it. You don't need a bunch of adjectives and adverbs. It's short and sweet. The big thing here too is after I read it, it should be clear to me that you understood what you did. Right? So when I'm reading it, if you have a bunch of mistakes, like if you get a lot of points for it should be accurate and correct. Not only do you miss points for each thing that you write that's incorrect, but then if it's enough of them, you're going to lose points for that too. Like it just seems like you got no idea what you're doing, you're going to lose points. Any questions about that? All right. So this is also a little bit about it. It's not Google Classroom, but there's a rubric I'm going to share with you. It's actually already out there. It's in WVSU Online, so you can go ahead and look at it now. But like I said, right, you're wondering how long does it need to be? And I don't want it to be long. I want it to be short. This rubric that looks just like this, I mean, it's longer, but this is just a little snippet of what it looks like. All you have to do is literally start at the top and work your way down. Is there a title? Oh yeah, I should give this thing a title. Is the title appropriate? Right? So if it says photosynthesis lab, you're gonna lose points for that. But if it says, how do different colors affect the rate of photosynthesis? That would be more appropriate if that's the lab you did. Is there an abstract section that's labeled? Right? All these things. You're going to have like I said, you're going to have this worksheet. Just go from the top to the bottom and make sure you check off all those things, and your grade should be fine. Here's the different sections. We're going to talk about it very quickly. This is how it's going to. Oh yeah, remind me. Let me back up too. When I said don't use the internet, now I remember why I wrote that. What I mean is this: do not Google how do I write a lab report, because there's so many different ways to write a lab report. And this is the way you need to write this lab report. Just like in a peer-reviewed journal. If a scientist does a study and they want to submit it to this journal and this journal and this journal, they're going to have to reformat it. This journal wants it this way, this journal wants it that way, this journal wants it this way. So my point is, again, do not use the internet, do not search, how do I write a lab report? Write it like I'm about to teach you. There we go. Here's the different sections. We're going to talk about it briefly by themselves. There's the title, the abstract, introduction, materials and methods, results, discussions, figures and tables, and literature cited. So let's jump into it. First of all, there's the title. I've already mentioned this. It's pretty straightforward. The title should reflect the experiment you did. So your title shouldn't be lab report. Your title shouldn't be photosynthesis respiration lab, right? Your title should describe the actual experiment that you did. It's pretty straightforward, right? Any questions about that? All right, here's the part that people get a little bit confused at, so I want to make sure you understand it. The next section after the title is the abstract. Abstract will be labeled. There'll be the word abstract. You're going to write a little bit. And this is a brief summary of what the paper is about. And in a sense, this is probably the most important thing for you as non-biology majors. Because like I said, hopefully as critically thinking citizens, one day you're going to be doing your own research and looking up other peer-reviewed journals. And generally speaking, when you're doing that kind of research, you're usually just reading the abstract, right? You're just usually looking like, all right, what's the basics of what they did? The basic experiment, not all the details, but here's basically what they did, and here's basically what the results were. Right? That's all you're going to do. So, again, if you did the one about light intensity, you would say, in this experiment, I was trying to see if light intensity affects the rate of photosynthesis. To do that, I put algae under different lighting conditions and measured the rate of photosynthesis and respiration. You don't even need to get into the pH and any of that yet. You just say that you put them under different light conditions, and I found out that yes, the algae in the most intense light photosynthesize the most. Or whatever it may be. So just the basics of what you did and the basics of your results. That's it. You don't even need to have any background yet, right? So again, like I just said now, you're going to tell them like, oh yeah, this one photosynthesized more. But you don't even need to get into the specifics and say, I know it photosynthesized more because I track the pH every 5 minutes, and this one increased more than the rest of them. You don't even need to say that. Just the basics. That's what the abstract is. Any questions about the abstract? All right, introduction. Now this, and it's kind of weird, in a sense you might think the introduction should be before the abstract. Let me tell you why. The introduction is the thing that's going to give the reader enough background information to understand everything, right? So later, not the introduction section, but in other sections, you're going to describe what you did. You're going to say, I put these things under the light, I check the pH every 5 minutes. Now, if somebody was reading what you did and only read that and didn't read the introduction, they might be, what? Why the hell are they looking at the pH when we're talking about photosynthesis? This is where the introduction comes in. You're going to explain why you're looking at the pH. You're going to explain the whole basics of photosynthesis and respiration, right? You're going to give enough information about photosynthesis and respiration that the reader will understand the rest of the lab report. So again, eventually in the introduction, you're going to talk about how increasing pH means it's photosynthesizing. You're going to talk about how decreasing pH means it's respiring. And before you can even get to that, you obviously need to, at the very least, explain how photosynthesis uses carbon dioxide and how respiration produces carbon dioxide, right? Now, when I had that little star next to do not use the internet, this is what the little star was for. You don't need to use the internet for this. All the information you need is technically in that lab worksheet and or in your textbook. But if you want to, you can use the internet to get even more information about photosynthesis and respiration. You don't need to, but you can if you want. So any questions about that so far? All right, so again, you're going to give, you should do it in this order. Give a little bit of a background about photosynthesis and respiration. Then, even though you already mentioned this in the abstract, you're going to mention it again. The question I was answering in this experiment was this, right? You're going to repeat the question. You're going to include your hypothesis. So, again, I wanted to see how light intensity affected photosynthesis. I hypothesized that the more intense the light, the more photosynthesized. And then you also have to have a little sentence about why you chose this. Oh, also, this is a good time to remind you too. The lab worksheet that you do, a lot of it is meant to be almost where you can copy and paste it, right? So you have to write your hypothesis in the lab worksheet. You have to have a sentence of why you chose that hypothesis. So you've already done it for the lab worksheet. When it comes time to write your lab report, just copy it and paste it. Again, like I mentioned earlier, the introduction, we're trying to give enough background information so that the rest of the lab report makes sense. So you need to explain how changing the pH affects, or how we use the change of pH to measure photosynthesis and respiration. Right? It should make complete sense. That way, when the reader later, when you're talking about your results and say, this one changed the most, the pH increased the most in this one, they won't be thinking, what does that mean? Because they'll already have read it in the introduction. Likewise, you never have to say it again. So when you're talking about your results, which we'll talk about later, your results section, when all you have to do is say the pH started here, it ended here. You don't have to say, well, that means it photosynthesized, because you've already explained it here. There's a slight exception to that rule. We'll get there in a second. Any questions so far? All right. There's a lot of things the introduction should not include. So a lot of times people try to put the results in there, and you don't do the results, right? That's in a different section. Do the basic results in the abstract, and then later in the lab report, which we'll talk about here shortly, you will describe your results. But for the introduction, don't put the results. All you do is say, just a background information. This is a little background about photosynthesis and respiration. Here's the question we're answering, or I'm answering. Here's my hypothesis. Here's why I think my hypothesis. And here's how the change in pH is used to measure photosynthesis and respiration. That's it for the introduction. Any questions about the introduction? All right, materials and methods. This is really short and sweet. This is just basically where you write down what you did. That's it. Just write down what you did. But you need to be detailed, right? So you can't just say, I put 3 different cuvettes under 3 different lighting conditions, and that was it, right? You can describe the fact that you measured the pH every 5 minutes. That you compared it, and to do that, you compared it to the pH chart, right? All that stuff. So you need to mention everything you used and everything you did. In short, like a good rule of thumb is somebody who reads it should be able to take what you wrote and do the experiment themselves. That's how detailed it should be. What you don't want to do is copy and paste the directions from the lab or the lab worksheet into your lab report. So at the very least, first of all, it's supposed to be first person past tense, which the lab manual is not. So don't copy and paste it. Also, I'll point this out, write what you did, not what the instructions said to do. Like I said, we might call some audibles, we might change some things up. The people who are doing the light experiment, we might add a fifth color. So in that case, when you're writing your lab report, don't write what the instructions said, which was the 4 colors, write what you actually did, which was the 5 colors. So any questions about materials and methods? All right, once again, some things that shouldn't be included. For the materials and methods, again, you're going to say, I measured the change in pH every 5 minutes. But because you already explained why in the introduction, you don't have to explain it again. You already explained it. And likewise, this is just materials and methods. So you don't talk about what the results were yet. You just say, this is what I did. You don't talk about your data yet. So any questions about materials and methods? All right, we're making good time. Results. You've been trained to do this already. I don't know if you've noticed. A lot of the labs you've done, after you've taken your data and there's a table, and you have your graph or whatever, then there's questions, and it seems redundant, but this is why I've been training you to do this. There's a question where it just asks you to list your results, right? Like, so think about the biological molecules. You said, all right, the one that had the least monosaccharides was this, followed by this, followed by this, followed by this, right? That was a test, that was a question where you just listed the results. You just described the data. And then after that, it said discuss your results. And then basically you just kind of repeated yourself, and now you were explaining whether your hypothesis was correct or not. Well, that's what's about to happen here. Results in the discussion section is what you've already been doing this entire semester. So the results section simply puts the data into words. So you're going to have your figures and tables. You're going to have your table, and you're just going to describe it in paragraph form. You're just going to write what it says in the table. The pH for the one that was in the intense light started at this, and it ended at this. The pH of the one that was in the dark started at this, and it ended at this. Or like if you're doing the one for the colors, right? You would say the pH for the one in the red light started at this, ended at this. It's pretty simple, right? You're going to just describe your results. And in some cases, well, no, not some cases, all cases, not only are you going to say, all right, this one started here, this one ended here, this one started here, this one ended here, this one started here, this one ended here. Now, it's a little bit redundant, but then after you've said all that, even though it's there, then you say, this one increased the most, or this one decreased the most, whatever the case is, right? So even though you've listed it, in paragraph form, then you'll also reiterate the fact by saying, this is the one that changed the most, or the one that changed the least. So any questions so far? All right, if something goes wrong, and one of them doesn't change, you're going to mention that too. Even though, again, to start off with the results, you're going to say started here, ended here, started here, ended here. And then once again, you're going to reiterate that important point and say this one did not change. Any questions? All right, another mistake I see a lot. So you're going to number these little things, cuvette number 1, cuvette number 2. Don't describe them like that in your results section. So just like I was mentioning earlier, you're going to describe them by the condition. The one in the green light started here, it ended here. The one in the red light started here, it ended here. So you're not going to say cuvette number 1, cuvette number 2, right? You're going to describe them by the condition they're in. The one in the ice started here, it ended here. The one at room temperature started here, ended here. So describe them by their condition, not by the number. Any questions about that? All right, here's where people screw up too. There's no need, don't even mention the word photosynthesis or respiration. Not in this section. Because in this section, it's nothing but the data. How did the pH change? That's it. So when you say the one, the one in the white light started here and ended here, and it changed the most, yes, that means photosynthesized the most, but you don't mention that yet. That comes later. So in this section, you just give the results. You don't talk about what the results mean yet. That is in this section. In the discussion, that's when you talk about what your results mean. So and it seems a little bit redundant, right? Because you just wrote down all your results. And then the very next thing you're going to do is explain what those results mean. To do that, the first thing you're going to need to do, and again, redundant, because you did it in the abstract, you did it in the introduction, you're doing it again. You're going to restate the purpose of the experiment. What question was I answering? Right? What was my hypothesis? And was it correct? That's basically what you're going to do, right? You're going to explain the results again. So like I said earlier, when you said, all right, the pH started here and ended here, that's it. You just talk about the pH in the results section. In the discussion, you explain what that means. So it started here and ended here. That means this is the one that photosynthesized the most, right? Any questions so far? Right. Again, you're going to restate the question. You're going to restate your hypothesis, even though you mentioned it before, right? You're going to restate your hypothesis, and then say, here's the results. This is what it means. Was my hypothesis correct or not? Or, like I mentioned a few minutes ago, if your data is inconclusive, like that one Petri dish experiment that you guys did last week, it's okay to say that. Like, this data is inconclusive, so I don't know if my hypothesis was incorrect or not. But if that happens, you do need to at least write a sentence or two where you at least guess what you think went wrong. Another thing, and you will lose a point for this. Do not use the word prove. So let's say your hypothesis was supported by your experiment. Do not say, after doing, here's what the data says, and it proves that my hypothesis was correct. It's not a proof to support your hypothesis, right? It's not. We've talked about this earlier this semester, right? Proof. We don't have enough data for it to prove anything. Any questions about that? All right. In the lab worksheet, as you've already done, you've been trained to do, it's going to ask you, what are some flaws in the experiment? The experiment. How can the experiment be approved? And you're going to have the same thing in the lab. Right? You've been trained to do this. I've been having you do this all semester. But once again, I'll reiterate, I'm talking about the experiment design, not the lab worksheet, not my way of presenting the information, not the material. Well, you may be able to talk about the material. For example, if VJ screws up again and doesn't bring all the materials in at first, I need to know that in general. That wasn't a good thing. But you don't need to write that in the lab report. I don't care about that. I'm not talking about how great the lab went. The experiment itself. If you were a real scientist trying to publish your findings, what could you do to make your experiment better? That's what that means. So any questions about that? All right, so let me see, what was this example? If results suggest algae photosynthesize better at more intense light, does this mean there's no limit to light intensity? Oh yeah, that's a good example, so you can't use that one, right? But you might say, if your hypothesis was that the more intense the light, the more it'll photosynthesize, and your results supported your hypothesis, but does that mean if you keep going that will always be true? Like, don't you think eventually there might be a light that's so bright that it would just stop working? That's just an example. But anyway, any questions about that? We're almost done. All right, and then finally, and again, you've been trained to do this too, discuss other experiments you could perform to test your hypothesis. Because again, in real science, you don't just test your hypothesis and you're done just because it was supported, right? You would go back to the drawing board and think about different ways you could test your hypothesis. I'll give you one example, and then you won't be able to use it. Rather than observing pH changes in the cuvettes for 45 minutes, which is what you did or going to do, you could put algae in 2 fish tanks with different amounts of light and observe how much it grows, right? So if you were looking at light intensity, you're doing that first experiment. That's one way you could also test your hypothesis. All right, I took a picture of it at the top of my tank. It was like this much algae, and then by the end of the experiment, there was this much algae. Whatever. Anyway, any questions about that? Okay, and again, I'll remind you, you've been trained to do this. All those seemingly redundant questions on the lab worksheets that you've been doing, this is why. It's been trying to get you ready to do this. All right, the next section. This is very, very simple. Figures and tables. This is simply where you're going to have your table, followed by your graph. Table, right? This is the very easiest part. You've got the table in your worksheet, so all you need to do is recreate it. Probably could even just copy and paste it. It's just the table. Copy it and paste it. Don't forget to label it properly, right? It should say table 1, colon, and then describe, give it a proper name. So you don't want to just leave it at table 1. You're going to talk about what the data is. Include your units. Of course, units in this case is just pH and time in minutes, right? 5 minutes, 15 minutes, so on and so forth. Any questions about the table? All right, the graph. This isn't even tricky, but it's technically trickier than the table. So far this semester, you've always had the option of using a piece of paper for your graph or doing it on a computer. This is the one time in the semester where you're gonna have to do it on a computer. I can help you do that if you want, so you're not on your own. But yeah, you're gonna need to do a graph. All the things I've been teaching you about graphs throughout the semester, just use those lessons. We're gonna start it with figure 1. Excuse me, let me make sure we're still recording. And record. Sorry about that. Where are we at? There we go. Yeah, make sure you use your units, right? Make sure your independent data is on the variables on the x-axis, and your dependent is on the x-axis, y-axis. Excuse me. So all the stuff you already know, right? Include your units. Yeah, nothing new there. Only new thing here, again, is that this is the one time that you're forced to use a computer, some sort of computer program, to make the graph instead of doing it by hand. Of course, for the lab itself, that's due next Friday, as usual, you can do one by hand. But when it comes time to turn in your lab report, if you did it by hand, you need to also do it by computer. Any questions about the figures and tables section? All right, this one's really easy. People still lose points for it because they just don't add it. Don't let the fact that this is so easy make you forget to do it, right? So you do need, at the very end, you need a literature cited section. It'll say literature cited, and you're gonna list your sources. And if your only source was your lab worksheet, then you just write that, lab worksheet. But again, if you look stuff up on the internet, you're gonna write, put a little link. I don't even care about the style, like APA format, I don't care. As long as it says literature cited, and you just in some way list all your sources, and if it's a source other than the textbook or the lab report, lab worksheet, it's any other source, I just need a link to your source. So if you fail to do that, you'll lose points, right? This is bigger for the other classes, the online classes, but yeah. Make sure you list your sources. There needs to be a working clickable link if you have a source other than the textbook or the lab worksheet itself. Any questions about that? Any questions about lab reports or next week's lab in general? All right, make sure you're on time. Don't be late. This is a team effort. When you're late, you're gonna come in, you're gonna slow other people down. Please be here on time. Next week, the door is gonna be locked and not open. At 9 a.m. it's locked, and I will not open it back up. So that's it. If you came in late, make sure you email me and tell me you came in late so you can get at least partial credit for being here today. I will be in office hours until noon if you need it.